So welcome. This session will be looking at insulin, and by the end of this session, you should know the structure of insulin, how it is synthesized, how its secretion is regulated, its metabolic effects on various tissues on the body, and its mechanism of action. Welcome all. My name is Wallace Munyeri, and I'm the Daktari. Welcome. Insulin is a peptide hormone which is produced by the beta cells of the islets of Langerhans. The islets of Langerhans constitute the endocrine portion of the pancreas. Okay, remember pancreas has both exocrine portion and the endocrine portion, so it is a compound gland. And within the islets of Langerhans, we also have other cell types: the alpha cells, the delta cells, the epsilon cells. But today we'll be looking at the beta cells specifically to produce insulin. Now, insulin, its metabolic effects generally we can say it has anabolic effects, but to some extent this is not entirely true, because it also promotes glycolysis. Okay, so when glycolysis, remember, is catabolic, but mainly it's anabolic in that it will promote glycogenesis both in the liver and the muscles, triacylglycerol synthesis in the adipocytes, protein synthesis in the liver and the muscle. Now, the structure of insulin. Insulin is composed of 51 amino acids, which are arranged in 2 polypeptide chains. Now, let me take that. This I'm drawing at the I'm trying to simulate the polypeptide chain. Now, this is chain A here, chain A, and chain B. Okay. Now, chain A has 21 amino acids, and this has that. Okay, and add them is 51. Now, they have intermolecular disulfide linkages. Now, the cysteine at position number 7 here will link with another cysteine at position number 7 here. Okay, so a disulfide linkage there. Then see another cysteine here at position number 20. Okay, we link with a cysteine at position number 19, the B chain, for another disulfide linkage. Then on the A chain, we have an intramolecular disulfide bond between the cysteine at position number 6 somewhere there. We started here 1, and another cysteine at position number 11. Okay, so there will be intramolecular disulfide linkage. Okay, if you remember that, I think you'll be with that. Yeah, it's like I've summarized this diagram here for you. Cysteine at position number 7 with another cysteine at position number 7, cysteine position number 20 on the A chain, cysteine number 19 on the B chain, then intramolecular disulfide linkage between cysteine number 6 and cysteine number 11 on the A chain. Okay. Now, synthesis of insulin. Now, we have 2 inactive precursors of insulin, that is pre-proinsulin and proinsulin. Okay, so which are usually cleaved to produce the active hormone, that is insulin. Okay, let me go back and show you something here. Now, this is pre-proinsulin. Can see pre-proinsulin has the signal sequence attached to the N terminal of it. Okay, then in the endoplasmic reticulum, the signal sequence is cleaved, and what we form is insulin here, insulin, and plus the connecting peptide or the C peptide, that is now proinsulin. Then within the Golgi apparatus, the C peptide is cleaved and we form insulin and the C peptide. I hope this has made it easy. Now, C peptide, is it important? Yes, because it ensures the proper folding of insulin. Okay, and another function, since this C peptide has a longer half-life in plasma than insulin, it can be used, its plasma levels can be used as an indicator. They can tell us how insulin production and secretion is. Okay, so you can see here it's essential for the proper insulin folding because its half-life in the plasma is longer than that of insulin. The C-peptide levels are a good indicator of insulin production and secretion. Now, insulin is usually degraded by the insulin degrading enzyme found in the liver and the kidney. Okay, but mostly in the liver. It has a very short half-life of 5 minutes, and so its duration of action is 20 minutes. Okay, so so remember it will undergo 4 half-lives, and once it undergoes 4 half-lives, it has been terminated. Now here is the synthesis. Now first we encode genes encoding for insulin are usually transcribed to a messenger RNA in the nucleus. Then the messenger RNA will move into the cytoplasm for translation, okay, which is initiated by the cytoplasmic ribosomes. Then before we start translating, there will be formation of an N-terminal hydrophobic signal sequence, okay, and then that N sequence will direct the messenger RNA together with the ribosomes to move to the left endoplasmic reticulum. Then once at the left endoplasmic reticulum, it will penetrate the membrane of the left endoplasmic reticulum. Then when we start elongation, or the translation of the polypeptide chain that will form insulin eventually. The polypeptide chain will usually form in the elongation of the polypeptide chain is directed into the lumen of the left endoplasmic reticulum and will form pre-proinsulin. Okay, then the N-terminal signal sequence is cleaved within the lumen of the left endoplasmic reticulum to form proinsulin, which is packed into vesicles and transported to the Golgi apparatus. Okay, once in the Golgi apparatus, the C-peptide is you can see here. The C-peptide is cleaved from the insulin and packed into vesicles. Okay, so in these secretory granules, you'll find the insulin and the C-peptide, and when there is a proper stimulus, it is secreted into it is exocytosed into the blood vessels. And remember, that is why we said you can use the C-peptide levels, the plasma C-peptide levels, as an indicator of the insulin production and secretion. Now, regulation of insulin secretion. Now, secretion is usually regulated by blood-borne hormones and fuels. Now, in which instances will we have increased secretion? Now, remember, first let's read these steps in that the insulin secretion by the beta cells is usually closely coordinated with the secretion of glucagon by the alpha cells, in that they are antagonistic. When you have high insulin levels, the glucagon levels are low. When you have high glucagon levels, insulin levels are low, so they are antagonistic hormones. Now, the relative levels of glucagon and insulin released are normally regulated such that the rate of hepatic glucose production is equal to the use of glucose by peripheral tissue. What does that statement mean? Now, if you have increased glucose usage by the peripheral tissue, say when you are running, okay, your muscles will take up the glucose, so there will be low glucose levels in the blood. So insulin secretion will be decreased, but glucagon will be increased so that we can replenish the blood glucose levels. Then when say you are not using the glucose, or after a meal, the blood glucose levels become very high. Okay, so you want to reduce these blood glucose levels. You will also, you can mainly you will affect the muscles, the liver, and the adipocytes to reduce these glucose levels. Maybe I'm going too far. We looked at when we are discussing the metabolic effects, but the blood glucose levels must be maintained between 40 and 140 milligrams per deciliter. Now, okay, but in particular, insulin secretion is increased by increased blood glucose levels, amino acids, and also we have other hormones known as the gastrointestinal peptide hormones, which you will see. Now, glucose how does glucose cause increased glucose levels cause insulin release from the beta cells? Now, ingestion of carbohydrate-rich meals leads to a rise in blood glucose. Now, which is the primary stimulus for insulin secretion. Now, the beta cells are the most important glucose sensing cells in the body. Okay. Now, like the beta cells, like the hepatocytes, also the beta cells contain the GLUT2 transporters, and they usually express glucokinase instead of hexokinase. Now, if you remember your glycolysis, we said glucokinase has a high Km, so it can only phosphorylate glucose when the concentration of glucose is very high, and like hexokinase, which has a low Km, so can phosphorylate glucose at even minimal concentration. So glucokinase is activated when there is high concentration of blood sugar, blood glucose. Okay, once activated, remember it will form glucose 6 phosphate. Then we continue with glycolysis. Okay, which will eventually lead to ATP generation. Now, leading to insulin secretion. But how will this ATP lead to insulin secretion? That is what I'm about to tell you. Now, the beta cells, now glucose dependent release of insulin into blood is mediated through the rise of calcium concentration in the beta cells. Now, let me make this simple. Maybe I should draw that. Then, so if here you have a beta cell, imagine this is your beta cell. So on its surface, it has ATP sensitive. This I've drawn here is an ATP sensitive potassium channel. Okay. Now, when blood glucose levels increase, glucokinase will act and ATP will form. Then ATP will cause closure of these potassium channels. Let me write K plus channels, K plus, it will cause closure. Once closed, it will lead to depolarization of the cell membrane, and once depolarized, there will be opening of the voltage gated calcium channels. Okay, so calcium channels will be there'll be opening, and there will be calcium influx. Calcium will move from the extracellular space into the cytoplasm. Now, calcium is the one which will cause the secretory granules. Now, this will cause the secretory granules. These are the secretory granules which contain your insulin and the C-peptide. Because once calcium enters, it will cause the exocytosis. So there will be, you will see release of insulin. Okay, that is what this statement is saying. Now, we have a clinical note. We have drugs, namely sulfonylureas and magnetide analogs, okay, which are used to treat type 2 diabetes, okay. How do they act? They usually block the calcium channels, and they will lead to insulin secretion. That is why these drugs, they are also known as you'd find them under a class known as insulin secretagogues. Okay, so it's like they are making the beta cells to produce insulin. Okay, I hope that point is clear. Now let's continue. Also, increased also how amino acids, how will they cause? Now, ingestion of protein causes a transient rise in plasma amino acid levels such as arginine that enhances the glucose-stimulated secretion of insulin. Also, fatty acids will have a similar effect. So, increased amino acid levels and increased fatty acids after a meal will cause the insulin secretion. Also, now, let's look at this one, gastrointestinal peptide hormones. Now, the intestinal peptides, glucagon-like, the intestinal peptides such as glucagon-like peptide, abbreviated as GLP-1, and also gastric inhibitory polypeptide, GIP, which is also known as the glucose-dependent insulinotropic peptide, usually increase the sensitivity of the beta cells to glucose. Okay, they are usually released this glucagon-like peptide is released by the L cells, and the GIP is released by the K cells of and as the name says, gastric, the stomach. Now they are usually released from the small intestine L cells. This one, the glucagon-like, after ingestion of food. Okay, so after ingest food, the stomach and the small intestine will release these gastrointestinal peptide hormones, causing an anticipatory rise in insulin levels. Okay, and these gastrointestinal peptide hormones are also known as sorry for this, are also known as incretins. Okay, okay, they are also known as incretins, and their action may account for the fact that when you give the same amount of glucose orally, there will be greater secretion of insulin than when you induce the same amount intravenously. Why? Because when you give the same amount of glucose orally, the gastrointestinal tract will produce these incretins. So there will be the beta cells will be more sensitized, unlike when you give it intravenously. Now, these gastrointestinal peptide hormones are usually degraded by an enzyme known as dipeptidase 4. Okay, now clinical note. We have drugs known as the GLP-1 agonist or analogs, and also others known as the DPP-4 inhibitors. Okay, and these are used in the incretin therapy, can be used as antihyperglycemic or the hypoglycemic drugs for patients suffering from type 2. Okay, examples of DPP-4 inhibitors may include sitagliptin, such drugs. Okay, but I hope you have understood how these drugs can work, because if you have GLP-1 agonists, they will sensitize the beta cells to glucose, and when you have DPP-4 inhibitors, they will inhibit DPP-4, the dipeptidase 4. So once you inhibit DPP-4, it will not degrade the gastric these hormones, the gastrointestinal peptide hormones. Okay. Now, decreased insulin production. When does one have decreased insulin production? Now, the synthesis and the release of insulin are decreased when there is scarcity of dietary fuels, such as when one is fasting. Also, during periods of physiologic stress, such as infection, hypoxia, also during times of vigorous exercises. Okay, which this physiological stress can lead to hypoglycemia. Now, to prevent hypoglycemia, we will reduce insulin production and increase glucagon production, or also we can increase the catecholamines. Now, these effects are usually mediated by catecholamines, namely norepinephrine and epinephrine, or noradrenaline and adrenaline, which are made from tyrosine in the sympathetic nervous system and in the adrenal medulla, and then secreted. Secretion is largely controlled by neurosignals. Okay, so there will be a role of the brain. So catecholamines, primarily epinephrine, has a direct effect on the energy metabolism, which causes a rapid mobilization of energy yielding fuels. Okay, so there will be a mobilization of glucose from the liver. Okay, so there will be glycogenolysis will be increased, so, and also gluconeogenesis will be increased. Also, fatty acids lipolysis will be increased in the adipose, so that to increase the fatty acids available. Okay. Also, in addition, these biogenic amines, namely the catecholamines, usually override the normal glucose-stimulated release of insulin. Thus, in emergency situations, the sympathetic nervous system largely replaces the plasma glucose concentration as the controlling influence over the beta cell secretion. Okay, so in emergency situation, the catecholamines are the ones which are acting. So catecholamines and the glucagon, it's like they have the same effect to increase blood sugar levels. Okay, so as to make glucose readily available if we want to run away from a danger, there'll be enough glucose for the muscles to do so. Now, there is an image. Insulin release is increased by glucose, amino acids, and fatty acids, decreased by the catecholamines. Now, metabolic effects. What are the effects of insulin on carbohydrate metabolism? Now, it usually affects three tissues, mainly liver, muscle, and adipose. Now, in the liver and the muscle, it will cause glycogen synthesis. Now, in the muscle and the adipose, it increases glucose uptake by increasing the number of glucose transporters. It usually increases the number of GLUT4 transporters which are expressed in the cell membrane of muscles and the adipose. Now, in the liver, insulin decreases the production of glucose by inhibiting glycogenolysis and gluconeogenesis. Also, insulin promotes the conversion of glucose to triacylglycerols in the liver. Okay. Also, then these triacylglycerols are secreted from the liver in form of very low density lipoproteins. Okay. Now, what are its effects on the lipid? It will reduce the release of fatty acids from the adipocytes. Okay. By inhibiting the hormone sensitive lipase, it will inhibit the triacylglycerol lipase. Okay. Also, it increases the transport and the metabolism of glucose into the adipocytes. Okay. Once in the adipocytes, this glucose usually form glycerol D phosphate. Remember from once you are doing glycolysis, remember dihydroxyacetone can form glycerol D phosphate. A reaction catalyzed by the glycerol D phosphate dehydrogenase, and then glycerol D phosphate triacylglycerol synthesis, because we will esterify fatty acids, okay, to the glycerol to form triacylglycerols. Now, what are the effects on protein? Insulin will stimulate both the entry of amino acids into cells and also protein synthesis. Here we are, after taking a meal, glucose, it will enter the liver, and you can see the entry into the liver is insulin insensitive. So you do not need insulin for the liver to take up glucose. Once in the liver, this glucose will mainly go to glycogen synthesis. Also, it can go to formation of fatty acids. Okay. Okay. Also, these amino acids, once in the liver, they usually go to protein synthesis. Okay. Also, in the muscle, the amino acids will form proteins. Then we have the fatty acids, which enter in form of chylomicrons. Now, the adipocyte, there will be expression of the lipoprotein lipase, which cleaves the very low density lipoproteins and chylomicrons to form fatty acids, which are esterified to glycerol to form triacylglycerol. Remember, now also glucose, you can see now this circle here is a GLUT4 transporter, so there will be increased uptake of glucose by the adipocytes, and this glucose can form glycerol D phosphate, which will be used in the synthesis of triacylglycerols. Also in the muscle, we have the GLUT4 transporters, and this glucose can either go to glycolysis and can also go to glycogen synthesis. Okay. I want you to note that the reason I put this is to show you that uptake of glucose by the liver and by the brain is insulin insensitive. Okay, so the brain and the liver they can take up glucose whether there is insulin or whether there is no insulin. But the muscles and the adipocytes, they are insulin sensitive. Now, mechanism. How does insulin act? We have insulin receptors. Insulin receptors are known as the tyrosine kinase receptors. The tyrosine kinase receptors are tetramers in that they will have 4 subunits, 2 alpha subunits and 2 beta subunits, with the beta subunit forming it has a transmembrane portion which is lipophilic and a cytosolic portion which is the tyrosine kinase. Once insulin attaches to the insulin binding site on the alpha subunits, causes conformation of changes which are then transmitted to the beta subunit, causing phosphorylation of tyrosine. Then that leads to the activation of the tyrosine kinase, which will phosphorylate another tyrosine, which is found in the insulin receptor substrates. Then, once those insulin receptor substrates are activated, they will go to activate multiple signaling pathways, okay, and which will lead to these metabolic effects. Increased glucose uptake, increased glycogen synthesis, increased protein synthesis, increased fat synthesis, but it will inhibit gluconeogenesis, glycogenolysis, and lipolysis. Okay, but that was just an image. Then membrane effects, especially in the adipocytes and the muscle cells. How does it cause the GLUT4 receptors to move to the membranes so as to increase glucose uptake? Now, glucose transport in some tissues such as muscles and adipocytes increases in presence of insulin. Okay, insulin usually promotes the movement of insulin-sensitive glucose transporters known as the GLUT4 transporters from a pool located in the intracellular vesicles to the cell membrane. Okay, note movement is as a result of the signaling cascade in which these now the insulin receptor substrate, after they are activated, they will bind and activate a kinase, the phosphorinositide 3 kinase. Now, the phosphorinositide 3 kinase will phosphorylate a membrane phospholipid known as phosphatidylinositol 4,5 bisphosphate to form phosphatidylinositol 3,4,5 triphosphate, abbreviated as PIP3, which will in turn bind to and activate the phosphoinositide dependent kinase 1. Okay, then after activating the phosphoinositide dependent kinase 1, it will activate AKT, or the protein kinase B. Okay, once that protein kinase B is activated, the GLUT4, the insulin sensitive glucose transporters GLUT4 will move from the intracellular pool to the cell membrane, causing increased uptake of glucose. Now, in contrast, other tissues that have insulin insensitive systems for glucose transport. That is why I said the hepatocytes, also the erythrocytes, also the cells of the nervous system, also the intestinal mucosa, the renal tubules, and the cornea do not require insulin for glucose uptake. Okay, unlike the muscles and the adipose, which require. Now, here's what I'm saying. Once insulin binds to the tyrosine kinase receptor, there'll be activation of the insulin receptor substrates, which will then activate for the kinase, which will in turn lead to the cleavage of a cell membrane phospholipid known as phospholipid in the phosphoinositol bisphosphate to form a PIP3. PIP3 will then there will be a signaling cascade until protein kinase B is activated, and this will lead to the movement of the glucose transporter GLUT4 from the intracellular pool towards the cell's membrane. Okay, and once in the cell membrane, there will be increased uptake of glucose. Then, after they have done that, when insulin levels decrease, the GLUT4 moves from the cell membrane to the intracellular storage pool, where it can then be recycled. But in other cases, they are usually degraded, okay, in the down regulation mechanism. Now, thank you for your attention, guys, and you have come to the end of that session. And please subscribe for more anatomy and biochemistry content. Thank you.